Good morning, everyone. My name is Jason Rodriguez. I'm one of the ROV pilots with Schmidt Ocean Institute. Today we are coming to you live from RV Falkor. I'd like to congratulate all y'all on all your hard work with MATE ROV Competition, World Competition. Today I am joined by two great doctors. I got Dr. Brennan Philips and Dr. Kakani Katija. Today they're going to be going all over their great systems that they've onto ROV Sebastian and going into detail and answering all your questions via the 4 systems. And you put your questions on the chat and they will relay them to me and I will ask for y'all. I'd like to start by going with Dr. Kakani and introduce yourself and where you're from.
Hey, my name is Kakani Katija, and I'm a principal engineer at MBARI, the Monterey Bay Aquarium Research Institute, which is based in Moss Landing, California. There, I lead the Bioinspiration Lab, where we develop a number of different technologies, including these imaging ones I'll be sharing with you, to really help us understand mechanics and movement of animals in the deep sea. That's my little spiel.
Yeah, and I'm Brennan Philips. Thanks so much for that awesome introduction. I'm at the University of Rhode Island, and my lab is called the Undersea Robotics and Imaging Laboratory, or URL. Yeah, we have all kinds of cool stuff that we've integrated onto Jason and his team's ROV, Sebastian here. So I think we're going to start at the front end, but maybe I should just quickly talk about what we're doing on this expedition.
That's a great idea, Brennan.
Because it'll lead right into your technology. So we're out at sea on this vessel, which is an amazing, sophisticated oceanographic research vessel that does all kinds of amazing missions. This is a special mission because we're exploring the midwater environment. We're not actually going to the bottom, although look at it. Most of the time, we're going and stopping the region between the surface and the bottom, looking at what may at first look like open water with nothing in there, but when you start to look a little bit closer, there's some amazing organisms to check out. Really delicate most of the time.
That's right. Mainly gelatinous, or they secrete mucus structures. So these are things that you can't actually collect using a net, which is a common or traditional way in which we would sample biological communities in the ocean.
Yeah.
Anything else? Oh, I should also add, part of the reason why this is really important, right, is it takes researchers about 10 years or so from the first discovery of an animal to describing it so that it becomes known to science. As part of this effort here, we're trying to really develop a much faster workflow so that we can get from discovery to description in a much, much shorter period of time. The reason why that's incredibly important is because about anywhere from 30 to 50% of animals in the ocean are yet to be described, according to some estimates from other researchers. If we're trying to, eventually flesh out our understanding of who lives in the ocean, we have to be able to speed up that workflow, which is all why we're here.
Do you want to move to the front? We'll start with Iris. Okay.
So there are 2 different imaging systems that we've brought on board from MBARI. The one here on the left is deep PIV, and then which stands for PIV stands for particle image velocimetry, which I'll get into a little bit. And then this one right here is IRIS, where RIS stands for remote imaging system. And these 2 systems together are being used to quantify not only the structures of the animals and what they're doing, either swimming or feeding, but also be used to measure things to like sub-millimeter precision, which is something that it's been really challenging to do robustly. The way we do this, these 2 different systems work is very, very different. This first system, Deep PIV, was actually integrated on the Falkor in 2019. So this is the second time, sorry, Sebastian. This is the second time that this vehicle has been out at sea here with the group here. The IRIS, though, this is the first time it's ever been integrated on Sebastian. Fortunately, though, this particular system has really just 1 housing, whereas DPIV has 1, 2, 3 housings that encase all of the components that are required. Both of these imaging systems, though, integrate onto the vehicle via a single interface can, which under normal circumstances would tend to keep water out. However, this entire cruise, we've been battling, trying to keep things dry. That's why you'll see a piece of tape there that says drain before use, because water doesn't work well. So here, the DPIV system is really comprised of a camera and a laser. There's additional auxiliary cameras as well, and that's really just used for positioning of the system. The laser, so the laser is here. The laser the laser remote head is here. The laser housing itself is massive, and it's located on the back here. So this housing itself has the 2-watt continuous red laser. I believe it's 671 nanometers. And that 2-watt red laser with a nominal beam diameter of about 1 millimeter gets injected into this fiber, which is approximately 10 microns in diameter. So going from 1 millimeter to 10 microns in diameter might not seem such a big issue if you're doing this stuff in the laboratory, but trying to do it at sea where you're maintaining these systems, you're trying to ensure that you're not losing any of that laser power as it's being injected. And also, while the vehicle gets batted around during launch and recovery, we have a lot of mounting systems in place to try and ensure that this connection is absolutely stable. Then here from the laser can, we just have the fiber optic cable, which then leads to the laser can over here, the remote head. And within the remote head, we have some optics that then turn that laser pointer light, right, into a laser sheet. For reference, a regular like laser pointer that you might be using for competitions or playing with your cat at home is roughly 10 to 30 milliwatts, whereas this is 2 watts. So there's a lot of power that goes into this, and it's so that we can very clearly illuminate the water in front of the camera. The optics, though, that are used is they create a light sheet. So instead of a single beam, right, we get a sheet of light, and then that sheet is then illuminating a plane in front of the camera. We can do things like measure particles that might be moving in the laser sheet, but the way in which we're using is for scanning. We can move this system in a laser sheet through an animal, especially if it's gelatinous or if a structure is made out of mucus. We can light up a number of planes or number of sheets within that structure, and like a CT or MRI scan, be able to reconstruct three-dimensional shapes. That's what we're doing here. We're trying to reconstruct the structures of gelatinous animals, as well as some of the mucus structures that they create. DPIV is used for scanning primarily gelatinous and mucus, things that we can relatively see through. Whereas IRIS takes a completely different approach. So IRIS is a single camera housing, so it's just one housing. Camera here, an auxiliary camera just to help with pilots positioning the instrument. And then we have this array around here. In this case, there's only 5 lights. Normal circumstances have about 6 or 7. But basically, these lights are on a hydraulic ram, which will then push out and then extend the lights outwards so that we have a lot of separation between our light and the image. This camera that's inside here is very, very different in terms of the optics that are used compared to the normal camera systems you might see on an RV. In this case, we're using this concept called planionic or light field imaging. What that is by integrating a micro lens array, so an array of tiny lenses in front of the camera sensor. What that does is it subdivides that single camera or imager into multiple imagers, then are focused on different locations within the volume. Therefore, that entire volume that we're imaging is in focus. More importantly, can measure in 3 dimensions, like the volumetric data about what is inside that field of view. We can do things like measuring surfaces of animal movement, let's say corals or sponges, as they're feeding. And as animals move, we can capture that movement with really high fidelity in terms of I think the dimension, the depth resolution scale is about 1 millimeter. The point is we can get very, very, very fine detail and then do measurements and dimensions so that we can understand length sizes. So this system, though, however, is used primarily for things that aren't transparent. So that then covers, the range of different animals we might be seeing in the ocean midwaters. Anyways, together with these two systems and the system that Brennan's going to be describing, we're hoping that this will then speed up the workflows that we need to try and describe life in the ocean.
All right.
Thank you. Yeah, so those are incredible imaging systems, and while they're so sophisticated, what I'm most impressed at is how quickly this workflow goes. So we're able to get up to some of these animals, approach them with the ROV with the assistance of our pilots who are super talented. And it takes sometimes, really what you're going for is a couple seconds almost, it seems like, right? When you get it in just the right spot, the frame rate of what your cameras are and the way it's all just operating and constantly streaming in. Once you get in the right spot, it only takes a couple seconds of just the perfect shot to get these really awesome, eventually 3D reconstructions and measurements that you can make of these animals. So it's really complex, but when it's actually in operation, this team has got it down so that they get up to these animals and get some really impressive information in a very short amount of time. So once that's all done, and all this wonderful imagery of these animals and information about it, then want to get a sample of it. The reason for that, again, coming back to the idea of describing animals in the deep ocean, that we always need to have a little bit of the tissue, at least, so that we can sequence its DNA and compare that with other animals that have been sequenced as well. Doing it this way because it's so difficult to grab the whole animal and get the actual holotype, as it's referred to, when you bring back the first of a new species, right? If you could do that with something that's very robust and cohesive, put it in a jar and you could do that. But some of these animals are like paper thin. You can't even imagine. They're mucus structures. So it's just an impossibility. What we've devised to handle that, I'm going to walk for a minute and flip with you. Thank you. Something called the rotary actuated dodecahedron, Rad Sampler. That's this system that you see over here. This system is really it's something on the product of about 4 to 5 years of development. Great. This is the product of something like 4 to 5 years of development with our collaborators at Harvard University and some other folks at CUNY Baruch College. So this system works kind of like this. It's better than describing it. It's 12 panels, hence the dodecahedron, that fold and encapsulate an animal like so. What's even more clever about this whole device is that it runs on just one motor. It's back here. So all it takes is this motor to spin. It's actually less than a full rotation. It's something on the order of 30 to 40 degrees. And when that turns, all these 5 arms turn well and translate that motion that they move together and encapsulate. Even incorporated some other cool mechanical parts to it, like these soft edges, which are 3D printed. That nice soft material that we're able to get out of there. And that gives a little bit of compliance. It's not a full seal when we close it. That's really, really hard, but it gets it pretty darn close and leaves just a few little orifices or some holes for a little bit of water to flow through. If we made it perfectly sealed, when we close up, the animal might actually get pushed out. So we encapsulate the animal like so. The camera that we put on the inside, so we get a little internal view. And then in here, we have a tissue sampler. So this, I'm going to take it off here. This is for safety, but has some sharp blades that we can control the speed. We can go slow and we can go fast, and we're able to get a little small tissue sample of that animal. Once we do that, there's a special hole. It's so hard to see right there where my finger is, right there. That little hole, ready to walk with me? Trace it all the way around the vehicle. Goes back in there, up the arm of the ROV, follow that tube, and all the way back out here to this system, which is on the back of the vehicle. And this is called a super sampler, SUPR. That acronym is sort of a legacy from what it was originally designed for. But what it does is it takes that tube inlet right there, and you can select one of 14 different places for that little piece of tissue to go. This is a rosette. So you can say, I want to go into port 6. Then goes back out this way after you've selected that, through these tubes to where my colleagues right here, get Becker, Dr. Kate Becker, Dr. John Burns from Bigelow Labs and Harvard University, respectively. And this filter stack that they're working on and preparing for a dive right now, there's 14 of those right there. They're sort of like wafers, sealed wafers, you can imagine that. So the pieces go through those tubes and into each one of those filters that we have right there. Once that's all done, back to this part over here, there's a bag. That bag is full of preservative. So we can collect the sample, get it onto the filter, and then inject this special preservative, which preserves the DNA intact in situ in the environment. That, again, happens within, well, our record right now is 2 minutes from collection to preservation. On top of that, all that amazing imagery that Dr. Katija's team has put together as well. So it's an incredible amount of information that we're gathering all at once. And then we can do that again. And we could do that up to 14 different samples on one single dive. Super impressive. So we've basically stacked this vehicle as much as we can, both sides, front, back, side to side, with all this amazing equipment. I guess the last thing I wanted to point out about this whole expedition and the whole thing we're doing here has nothing to do with what we've brought on board, because we're only here for 10 days. We rely on folks like J-Rod, J-Rod and his team make this vehicle work and make it click for us every single day. So, and we can't take that for granted. I mean, these systems are sophisticated enough, and we have our own issues, of course. We have to troubleshoot those all the time. Plenty of issues, because they're all new. But behind all that is this amazing vehicle that works for us every single day. A huge team on board to do that.
Full team all the way up from and all the way down. It's a big team that we have.
True. Yeah. So it's a real symphony almost, I like to refer it to, for everybody to come together and make this whole thing work the way it does.
Awesome. That was great. And now we ask y'all to type in your questions and send them in. And Connie. Okay. We have a question from YouTube. So
we're moving here.
Let's start with Kakani. Is there anything that helped inspire the designs of your two systems?
Right. Well, so in terms of the deep PIV instrument, there's technique that uses laser sheet illumination is pretty common in laboratory environments, and that's kind of the world that I came from in terms of using this kind of system to measure things like fluid motion, fluid mechanics studies. And so it was, I would say, almost accidental that this system, we discovered we could use it for scanning. Originally, it was just supposed to be used to measure particle movement, suspended particles that might be in water. And so that taking the ocean into the laboratory was really kind of the inspiration, the original inspiration for DPIV. But in the case of iris, the fact that insects have compound eyes, right? Multiple lenses that make up a single eye. That is kind of what inspired planoptic imaging or light field imaging. Oh. Being able to essentially get a volumetric image is the goal here, and that's inspired by insects. So very fun, a very fun fact.
Is this the first time that kind of camera system has been there, or used it somewhere else on the land-based?
So this imaging system, I mean, it's been around in the literature for several decades now. But in terms of using it at sea in the ocean, I am not aware of very many groups actually having done that, especially on an ROV. As far as I know, this is the only system that does that. But the system itself is generally used in industrial applications, like for inspecting parts and things like that. And so what we've been able to do is take a system that works just fine in a laboratory environment or somewhere in a warehouse, and instead package everything and then use it subsea.
That is great. Was there anything that inspired for?
Sure. I mean, I love this story. So previously, before I worked at URI, I was at Harvard University with Dr. Becker, who was right behind us on board on the ship, along with another person, Z. Ern To, who's another postdoc. He was a postdoc at the time, I think. He is an origami roboticist. there's probably like, I don't know, a handful in the world like that. But he's brilliant. Like it's a level up mechanical engineer who has this amazing way to look at structures and shapes and folding designs. And he had a concept of a folding icosahedron, 7-sided, solid, that he showed in a lab meeting one day when we were all together. It was really tiny. It was like centimeter scale type thing. Like, look at this cool thing I made. It folds up. And I go, should find a way to use that to capture animals underwater. Sounds like wow, but that's where my mind went because I have some background with the midwater stuff. But we as a team worked together to kind of scale it up. You know, Z really leading the design on that, but you know, a lot of input of how to make it work underwater. The idea of having compliance and some soft edges brought into that. You know, everybody kind of pitched in to make that all work as a prototype with you and the team at MBARI. It's a real privilege to be able to work with these midwater experts. We had this cool tool. We tested it. And now, with the support of the Schmidt Ocean Institute, we're here today in 2021 with the Rad 2, which is its fully blown version at this point. You know, actually large functional size. It has the sample collection ability, integrated cameras, and all that. It's not a fully 3D printed part. We actually made it out of real good, tough material that goes into it. So it took a number of years to get from this, like, it was like laser cut out of wood, all the way up to this structure that we see here.
Phenomenal. Yeah. There's lots of people that are behind the scenes working on all these technologies, and can't thank them all, unfortunately.
Have any more questions?
Oh.
Well, you have to repeat it, right?
Yeah.
Yep.
So they're asking, how many iterations of these cameras have y'all gone through before y'all actually got into the real world application of them?
So many. So DPIV, the laser imaging system, we actually started developing the system in 2015. In 2015, actually within 6 months, though, we were able to have a prototype. So we relied on the camera, the science camera, on the mini ROV, which is a smaller vehicle that we have at the institution, and then just used or deployed a laser housing in front of the vehicle. And we were just trying to see, okay, can an ROV be a stable enough platform to do this kind of imaging? Do we have the fine scale maneuverability or controllability of a vehicle that we need in order to be able to do this? And shockingly, it worked. And so then after that point, so that was version 1. Version 2 then involved a separate housing or integrating a separate camera housing so that you have a standalone measurement system. Version 3 involved incorporating another camera that was low light, very sensitive to low light, higher resolution, but unfortunately didn't meet all of our needs. And then version 4 involved a new camera system, but also a big laser upgrade because we first started with, around a watt, 150 milliwatt powered laser, and we always need more power, right? And so that we've integrated this 2-watt system. So since then, there's been a number of other components. So that, I mean, at least if I were to label something, it's been about 4 big iterations or redesigns of the system to get what we have today. And with Iris, this is actually the first version of the system.
Wow.
So
That's impressive.
We've been very fortunate.
Some lessons learned maybe with DPIV.
We've learned a lot. We also have some incredible engineers on the project, led by Paul Roberts at MBARI. Basically, yes, this is the first version. We've developed and built this ROV-based system. We're actually working on in parallel is a 7-camera system to go on an autonomous underwater vehicle. Can do kind of this volumetric imaging. And then hoping in the future to go off and do like 16 for vehicles where you're integrating imaging to vehicle nav, but also for animal imaging. This is just the beginning.
Awesome.
Cool.
All right. All right. So were there any special things that y'all had to design to be able to integrate your tools to SuBastian?
Oh, for sure. Yeah, I'll take it. Yeah, because like it's not like we walked on board and opened up some boxes and said, fellas, let's put this together now. No, we've been spending up like 2 years at this point in development to get to this point, iterating with people like Jason Rodriguez and his team and other folks that work at Schmidt-Ocean Institute to figure out, does it fit? How much does it weigh? How are we going to actually get your communication, which is really high bandwidth?
Yeah.
You know, how are we going to
dark fiber?
Right, exactly. How are we going to get that up through the ROV? And then the same thing for the Rad Sampler, because there's a lot of mechanical parts that are involved with that. So it took a long time, and it's been a very iterative process to the point where we showed up and it was like 4 hours and we were almost ready to go.
Unreal.
Yeah,
that does not happen.
Yeah, it was really cool. So like everybody did their job. It was perfect.
Awesome. We got another question right here. Why use Rad 2 to collect DNA rather than collect the whole animal?
It's a great question. I mean, you can do it both ways. I have to fully admit that there are plenty of other in situ. Yeah, so there are plenty of other ways like jars and suction samplers that have been used in the past. The Rad Sampler is just another way of that. It's like another tool in the kit that goes with it. I think the advantage of doing it this way is we're able to encapsulate with the same device different animals and then put them into these different ports in the back end. So we don't have 14 different jars up front, which takes up a lot of space and all that sort of thing. There's just one encapsulation chamber, and that gets ported out to these 14 different chambers on board. And then another question I'll just sort of anticipate it is, well, do we have cross-contamination that goes with it? We have on board, believe it or not, a qPCR sequencing machine that Dr. John Burns is running for us for some quality control, and it's been pretty good, actually. Things have been separated pretty well between the samples. That was one thing we also were concerned about, and we are able to do some certain flushing steps and all that sort of thing, and everything seems pretty good.
Awesome. I got another question right here. When is Kakani going to volunteer for a MATE ROV competition?
Ooh, on the spot.
Hi. I guess when I get home.
That's a good one. All right. How did y'all all get to become shipboard scientists?
Different paths for everybody, I think.
Yes, very different.
Why don't you go first?
Where do I start? So actually, funnily enough, I was a very competitive figure skater, and at the same time was just really into science and math. I was just like, cool. Fortunately, kept up with my studies as I was still competing, but decided to, well, in my case, I was like, oh, I got to get a job after I go to undergrad. Like, I need a degree that will ensure that I'll get a job, which is part of the reason why I went into engineering. I went to aerospace engineering. So I got a degree, an undergraduate degree in aerospace engineering, and then continued on to graduate school at Caltech in aeronautics. Then the joke I always say is that I got lost along the way. Instead of focusing on the stars, now focusing in the ocean. It's partly because what we're doing is we're exploring, we're discovering, and we're doing it now, not 5 to 10 years from now when we have a robotic system that can go out to outer space or carry people. And so that's essentially kind of what got my start. I started doing research on jellyfish, of all things, trying to understand how they propel and move for bio-inspired design. Like, what can we learn from them to maybe create more efficient or effective underwater propulsion systems? Right. And then very soon realized that, there was a very, very, very big need for tools, for us to understand how these animals function in their natural environment. That is really kind of what my group is focused on. I know Brennan is also focused on very similar questions. And that's how, got us here. And Brennan and I, gosh, we were talking, was it 2017?
Something like that.
Yeah, when we sat down, we're like, we should put our heads together, come up with a really great idea and try to get out here with Schmidt. And, fortunately now 2021, we've been able to do that now twice.
How about you, J-Rod? You started off in the Navy, right?
Right. Yes, I joined the Navy. I was in for 8 years as an aviation electronic technician. I was looking for an engineering job, and happened to be a bad time for the market in 2000. I ended up stumbling into ROV industry, company that worked to get veterans put into the oil and gas industry. And I got put into this, and I did oil and gas for about 7 years, and then I got invited here to Schmidt, and I've been here for 4 years, and it's been an amazing journey. I'm very privileged to be here.
Glad you're here too, Jared.
Yeah.
And yourself?
Oh, in a nutshell, really quick. I mean, I have to say I kind of always wanted to get here, so I'm like the kid who got his dream. I really love to go to sea. I kind of knew coming out of high school that I kind of wanted to get into ocean engineering. And plug for University of Rhode Island, we have an ocean engineering program. So that's like perfect. It's like engineering, ocean, let's do it. So I did all that, and then managed to get an internship working with Dr. Robert Ballard, because he's at University of Rhode Island. So kind of moved up through the ranks through that.
I have a question here. How easy or difficult is it to adapt new tech to Sebastian? Like more so like going between different ROVs, like to make it adaptable for the different ROVs that y'all work with.
That's true. I mean, your systems have to work on at least 2 different ROVs at Mbari.
3, 4, or yeah, three at Mbari. 3 at Mbari. Again, nothing is foolproof.
Right.
But then here, working with Sebastian. But you can take that question first if you want.
Well, sure. I mean, every ROV is different. I would say in the past few years, I feel a little bit of convergence. I mean, there's a number of science ROVs out there, at least in that category, science work class ROVs like Sebastian. We all seem to want fiber optic connections. We all seem to want to have some substantial power.
Mm-hmm.
Substantial.
Yep. And Ethernet is really important. So like if you have those flavors these days, that's sort of, we kind of like design our systems to interface with that type of thing. At that point, it's just like, is it the right style of connector and that sort of thing? I'm oversimplifying, but it does seem to be kind of coming down to that. I think a decade ago, when I was sort of early on in this, it was a lot more variant, like talks with this type of serial communication and that, and maybe several of them. Everything seems to be sort of converging towards that, which is really nice.
And so is your plan for it to eventually be commonplace for these systems to be on all research ROVs or AUVs?
We have to. There's so much time there's a lot of time that's spent in developing these new systems, but I can't tell you how much time, though, is sunk into integration, right? How are you going to get the system to work on all these different vehicles? So yes, we absolutely do need to work towards a standard interface for the community in order to speed up that process, because it's really, it's useful time, it's valuable time. Solve that common interface problem, then we've solved issues that groups can put on.
Let's see, what else we have here? Of course, the normal question, what's the coolest organism that you've encountered?
It's tough. It's always a hard question. I know, it stumps me.
You've ever encountered? I have a favorite.
Your favorite?
Giant larvaceans, because they make snot palaces.
And we've been on the hunt for them?
Yes, we have. Yes, we have. That's right.
Yeah.
And for you?
Oh, yeah. So every day I'm going to have a new animal. Today, my favorite deep sea animal is Tomopterus.
Yeah.
They're very cool.
It's a pelagic worm, but like you think worm, like ooh, worm. This thing is beautiful, amazingly beautiful animal. I Google, Tomopterus, T-E-R-I-S. Okay,
got it.
You'll find it. Something will correct me, hopefully, on that one. But they're an amazing animal. And last night we encountered a big, beautiful Tomopterus. And then I had to stand back and remark on they have 3 different types of bioluminescence. They make different colors, right? They drop bombs sometimes to countermeasures to get predators away from them. They swim is really cool.
Oh yeah.
Yeah, they can swim forward, stop on a dime, and reverse without changing their body orientation or anything like that.
And in theory, so can an ROV, but
nope.
They make it look a lot easier.
They make it look a lot easier. Yeah.
All right. Let's see if there's any more questions that we can.
Well, you had one about depth.
So
Sebastian is rated to
4,500 meters.
Right.
And that's primarily due to our flotation and our main control cans. A lot of the systems on board, like the 7 functions, actually are rated down to 7,500 meters.
And then DPIV and IRIS are rated to 4,000 meters.
And we got another question. Have we ever come across a giant squid? Have you all ever seen one? I have not.
I have not personally, no.
They are quite elusive. As far as I know, there's only 1 or 2 teams, I think, in the world that have ever actually seen and recorded giant squid in the wild. It has not been on this boat, but that's pretty hard to do.
We've seen some really big squid, but they're not proper giant squid. Like, I mean, these things are as big as me, but it's still not big enough to be classified as the giant. So there's a question for me, and it's what are the piloting challenges using the systems that y'all have brought on board? And there have been multiple, actually, because they all work different, and they all require a little bit different piloting techniques to be able to get the samples that they are wanting. With DPIV, though it's a wider field of view, the fine movements that we have to make with a vehicle that's as big as a car is very hard, but I mean, all we have to do is just slowly move through it, which makes it sound easy, but it's not. It's still really challenging. And then once you get that dialed in, then they want you to go to Iris, which then has a much narrower field of view and a really narrow focus point. So you can't see the animal inside of it when it's too far away. So trying to get it lined up is a really big challenge. And then when we come over to the Rad Sampler, we've noticed that with the color of it, although it's needed so that they can illuminate it and they can see the image on the inside, a lot of these translucent animals blend in with it.
They do, yeah.
And so
I didn't think of that.
we're using not only our cameras, but we're also using some of their cameras at times to give us as many different viewpoints as possible to get that 3D depth perception, to be able to get it right where it needs to be without touching the animal, and then they can encapsulate it. It has been a challenge. We're slowly getting better, I think.
Yeah?
Well, I'm curious, what's the hardest one out of all 3?
Ooh.
For myself, I would have to say Iris is the most challenging. And for
none of this surprises me.
And Cody thinks DPIV is more challenging than I. So I mean, we each have our own skills and what we're better at, and I mean, so
yeah.
This one, first time I did Rad, I was like, oh my god, this is going to be really difficult to do this,
sure.
And then one in a couple of days now.
You scored last night. That was an amazing catch.
Swaptress is not easy to capture. Yes.
Yeah. So have y'all ever thought about using augmented reality for using your systems and capturing animals?
Gotta take that.
Okay, I'll take that question. Short answer, yes. So at MBARI, a team of engineers led by Ben Irwin and Eric Martin have been developing a VR, a virtual reality imaging system. It's a 2-camera system that can then be integrated on the front of the vehicle, rated to 4,000 meters. But it allows us to get kind of real-time stereoscopic views of what's going on underwater. So you can wear a VR headset and fly the vehicle. We've also done some demonstrations using the VR headset and the camera system on our smaller vehicle, mini ROV, in our test tank facility. And then through, I think we had like 10 to 15 ROV pilots, including myself, try and do these comparisons between, using the traditional view with just the single camera versus the, wearing the headset versus, there's a stereoscopic view versus, just flattening 180-degree view, and comparing how well we're able to capture something in midwater. So we'd have like a cylinder that was attached to the vehicle, and we'd have things hanging in the test tank, and we'd have to compare like how quickly could we capture something with a vehicle with these different displays. And hands down, statistically significant, the VR headset stereoscopic view sped up the process for all of the ROV pilots that were part of the study. So we're hoping if we come back out again that we could bring that system and then you guys can also play with it.
Absolutely.
So cool.
How did y'all compensate for the vessel roll that we are currently doing right here with those VR headsets?
Well, So that's a really good question. And actually what we're finding, it isn't such a big deal if you have the optics right. And so because there's, a lot of focus on making sure you have the correct intraocular distance between the two cameras, if you have that in a really good state, and then it doesn't create this offending environment within the headset for someone.
Okay.
So in our case, nobody has gotten sick using the VR headset at sea while flying.
Okay.
But I can't speak to everyone because this is a work in progress. And so that project's only been around for 2 years.
Doesn't it depend on how rough it is?
In general. In general. You could say that for anything.
I know.
So do your jobs involve more science or more engineering?
No, this is an interesting question because I think a lot of the engineering we do, because it hasn't been done before, it really kind of fits in the science realm, right? We are testing hypotheses. We are trying to make things work. We're evaluating how things work and how we have to change them later on. So now, for me, at least, the work that we do, it's a bit mixed, right? There's that distinction between the two is a little bit difficult to make. I don't know about your...
No, I have the same answer. I don't want to be put in a corner on it. I think you have to be both an engineer and a scientist to do this type of technology development and testing. You have to wear both hats. And so, I mean, in my case, I have degrees in both of those, and I guess yours as well, right? In engineering and science. So you have to have both sets ready to go. It's hard.
Like even for me as a ROV pilot, getting the privilege to work with all the different scientists that I have, I've been able to learn a lot about the different organisms and their names, the difference between a ctenophore and a lobate ctenophore and a larvation house, which at first glance, some ROV pilots would think that was the same thing. But when the scientists are saying, ooh, can we look at that siphonophore? I know exactly what they're talking about in the screen, opposed to when I first started out. It was a big challenge, and so us kind of learning the whole science of it is making my job easier to give y'all the end product that y'all want.
It's all integrated. Yeah, we all have to understand what everybody else is doing.
So I get
the question. You have to say the question.
So I get this question a lot.
Sorry.
It's how can you become an ROV pilot and
take your job
and take my job? So taking my job's easy. Like, you just have to come and be better than me, and that's no problem. But to become an ROV pilot is actually a little bit more trickier. Almost every ROV pilot that I've had the privilege of working with came from different routes. There is ROV schools that you can go to, and they do a great job of teaching you the fundamentals, but they won't teach you ROV specifics because they're all so different. A lot of the work that we do is on the job training. But I would say trying to learn multiple disciplines. There used to be a term called a Renaissance man. Renaissance person is more what you need to be an ROV pilot, and that means that you need to know. You don't need to be an expert on anything, but you need to know a lot about everything. So hydraulics, mechanical engineering, electrical engineering, all these are big role into being an ROV pilot, because if you break it, you have to fix it. So when you're flying it, you have to also remember that if you're pushing things to the limit, that you're going to have to go in there and fix it. And a lot of these things, like the T4 itself, is full of thousands of different seals. I mean, legitimately thousands of seals in there. And getting in there and changing them, one little nick, one thing out of torque, arm is not working properly. So learning all these things, taking things apart while you're younger and putting them back together, and just understanding how things work together, hydraulics and electronics together, really help you get here. And then all you have to do is found out and yourself, and it's up.
We have a couple challenges.
Yes.
We'd provide.
Yes. You're not going to, well, I mean, it's possible that you could come out here and automatically be fantastic at flying systems that they brought on here, but the chances are not. years to finesse your skills at piloting a machine this big.
I mean, speaking to the MATE group, just if I could also add on to this discussion, because I understand that there are probably thousands of students who are interested in becoming ROV pilots or ROV engineers or some part of that putting in right now. And I think we represent that there are many different paths to get here. And that's normal. So there are formal ROV training schools. Mm-hmm. Some of them are operated by big companies like Oceaneering, for example. Have formal schools that they have work through and work within that company to move up. So there's that pathway. There's Memorial University in Canada, for example, has, I think it's a 2 or maybe it's also a 4-year program that gets you just for ROV piloting and with it. But you can also come in from an engineering or a physics background into places like Woods Hole Oceanographic or MBARI and that sort of thing and come at it from that angle. So, and there's no right or wrong way to that. At the end, we end up on the boat together. Exactly. So, yeah, I think it's a hard question to answer because there's no one straightforward way.
I mean, I think really it's, if you have a passion to do it and you're willing to learn what you need, and especially, electronic systems or mechanical systems and that sort of thing, you could get there. But then participating in research cruises or reaching out to researchers or engineers to see if there are opportunities for either internships or other training is definitely very valuable.
One of those people that somebody tells you that's not possible, and everything in your power to make it possible,
probably get it.
He wants you.
Yep.
Okay, so there's a question for me asking if, because we spent a lot of time off the coast of Australia, circumnavigated the whole country, and if I prefer the Australian waters to the California waters. In my experience, once we get 100 miles out, the ocean's the ocean, and especially when we're in the Pacific, I see a lot of the same thing. I mean, we see a lot of
soul misses.
Soul misses is everywhere. There hasn't been an ROV dive, I think, that I've ever done. I have not seen soul misses. But I would have to say Australian waters were very diverse. From the deep, cold Antarctic waters that were coming up and cup corals that we were seeing, and then up into the Great Barrier Reef, which, I mean, the Great Barrier Reef, being an ROV pilot on the Great Barrier Reef and one of the first to ever fly on it it's a privilege and an honor, and it's amazing.
It's pretty unreal.
Just the whole thought of it and realizing how big it is it's breathtaking. And I mean, there were a lot of massive discoveries in Australian waters because they have not had a lot of ROVs over there. We were one of the first to be able to do all the stuff that we had done. So there were a lot of firsts. I mean, first time a ram horde squid had ever been seen, the first time that we got a rock sample from what built the Great Barrier Reef. So a little biased towards Australia to answer the question. Unfortunately, I mean, and that's just due to the fact that the California waters have been pretty extensively, but not to say that they're not still beautiful and I don't enjoy coming out here.
Answer, J-Rod.
So there's a question about if y'all ever thought about joining some of the bigger ships like the Sir David Attenborough or some of the other bigger research ships. A little confused as to where they're going with that, but I guess, have y'all ever been on any of the other bigger ships?
I've sailed on a bunch of different boats as well. I mean, it depends. It's mission specific. Right. I don't know. There's.
Yeah, I mean,
if there's science to be done and it could be, helped along by any of our instruments, then you're totally open to it.
And I mean, we're upgrading the Falkor to the Falkor 2, which will be significantly bigger than this, and hopefully y'all writing your proposals already.
Yep.
And I have no idea what that one was.
That's okay.
So are there any more questions coming in? Let's see. I don't see any on here.
Great.
Okay. right. All right. So I guess there are no more questions. Is there anything that y'all can think of that y'all might have missed on your systems that y'all want to explain a little bit more, or?
No, I mean, I think you can actually quick Google search and learn a little bit more about the systems that we've put on. There's some information about all these systems actually. And again, if you have any questions, reach out.
Stay tuned because we're going to be in the water, this afternoon. What are we at?
In about 2 hours.
In 2 hours.
Okay, yes, we have work to do.
Yeah. Okay. And so are you going to be a part of MATE as well?
Oh, of course.
Yeah.
MATE, I believe it or not, hey, Jill, I go way back to one of the first competitions, international and regional competitions in the early 2000s. So MATE is near and dear to my heart. We have a team at the University of Rhode Island that competes every year in the MATE ROV competition, who I help advise. So I'm still involved. I need to get back. It seems like once I got going to sea, it was always on the same date as the competition. But that hasn't been every year. So I need to come back to it. And like you and me, let's go.
I want to get there as well.
And you got called in. And all of us.
I totally got called out.
And supposedly you something about a bike that you have to return to somebody.
Yep, there's something like that. Yeah.
All right.
Competition is awesome. I'm so glad that you guys are all part of that.
Yeah.
It really helped get me here. And there's so much to that community that enriches us. I mean, I know a number of people who've come through the MATE intern program who are now seagoing folks, marine techs and ROV pilots, and some who have gone on to techs and all that sort of thing. But the MATE program is integral.
I've been wanting to go so bad, like in the same thing. I've always been out here and never had the opportunity to make it out there, and really want to go see what these young minds are going to be bringing us. All right. So, let's wrap it up. Thank you all for joining us. Again, congratulations on all the hard work that y'all are doing out there. And join us in about 2 hours and 10 minutes for dive 459.
459,
Second
I think
second
that's
to last
it.
dive.
Second to last dive.
All right. All right, everybody.